Hi. First of all, thank you for this opportunity. My name is Bailey Delkamp, and today I'm going to be talking about my research on the tumorigenic potential of part and polyfluoroalkyl substances and chemoprevention by natural compounds. So first, a little bit of background information on why you should care about PFAS. PFAS are commercially utilized for their water-resistant properties and are otherwise known as forever chemicals. This is because they bioaccumulate in the environment and remain in the body for years. There are several exposure risks we have. You can be exposed to contaminated drinking water, especially in areas near industrial and waste sites. You can also consume fish from impacted areas. PFAS can leach into your food from containers and also be transferred through breast milk and house dust. As for the adverse human health effects that we see, a lot of the times it's increased cholesterol, increased uric acid, decreased birth weight, thyroid disease, and a handful of other ailments. As I said before, PFAS are known as forever chemicals. This is because of their carbon-fluorine chains that make them incredibly unreactive in the environment. Today we're going to be talking about two legacy PFAS. These are PFAS that have been used widespread in industry. They are perfluorooctanoic acid, PFOA, and perfluorooctane sulfonic acid, PFOS. I also decided to include a map of all the PFAS contamination sites in the United States that we know of at this time, just to reaffirm the fact that this is a problem for Michigan specifically, as we can see in the image provided by the Environmental Working Group. So when we want to think about how PFAS is causing all of these adverse human health effects, we need to look at its tumorigenic potential. This is measured as a function of gap junction intercellular communication, I might refer to as GJIC. Gap junctions are cellular ion transport channels. They are present in all signal transduction pathways, which are required for uncontrolled cell proliferation, a common phenotype of cancer. Improper functioning of gap junction intercellular communication poses a tumorigenic threat. On your left, you can see what gap junctions might look like when they're open. On your right, you can see what gap junctions look like when they are closed. My work builds off of the work of my PI, Brad Upham. He did a study on the in vitro effects of perfluorocarboxylic acids on GJIC. As there are over 15,000 PFAS compounds and derivatives, they usually are classified based on the length of their carbon chains, which vary from 2 to 10. And work by my PI showed that carbon chain lengths of 2 to 5 do not inhibit gap junction intercellular communication. Meanwhile, carbon chain lengths of 7 to 10 do. What is unknown, however, are the effects of the 6 carbon chain length PFAS. So that's where my first experimental question came in. And that is, does perfluorohexane sulfonic acid and perfluorohexane carboxylic acid, each of which are 6 carbon PFAS, inhibit gap junction intercellular communication? So to assess this, I used the scalpel load dye transfer assay, which involves loading Lucifer yellow as a fluorescence tracker along an incision line. In areas where gap junctions are open, the dye will diffuse into the surrounding cells, which you can see on the left as in our control. However, on the right, you can see PFAS-treated cells where the gap junctions are closed. Even though we loaded the Lucifer yellow onto the first line of cells through the incision, because the gap junctions are closed due to the PFAS treatment, the dye will not diffuse to surrounding cells. We also use ImageJ to measure the area of the dye diffusion and quantify it as a fraction of the control. So these are the results from our 6 carbon and 8 carbon chain PFAS and their ability to close gap junction channels. You can see that we did 3 doses. I want to draw your attention to the far right at our 75 micromolar dose. You can see that perfluorohexanoic acid, this third bar, was not very effective in closing gap junction channels, especially in comparison to the black bar, which is PFOA, its 8 carbon counterpart. This pattern also translates for the perfluorohexane sulfonic acid and its 8 carbon counterpart, PFOS. We see that perfluorohexane sulfonic acid does inhibit a decent amount of the gap junctions, but not nearly as much as PFOS. This aligns with the work done by my PI, as he had determined that the shorter the carbon chain length, the less effective it is in closing gap junctions. So then I was interested in studying the exact mechanism in which PFAS are able to close these gap junction channels, and I was able to conclude that perfluorohexane sulfonic acid inhibits GJIC via the protein kinase A pathway. To do this, I used H89, which is a potent inhibitor of the protein kinase A pathway, otherwise known as PKA. And we saw that we have our control on our left, and then our PFAS-treated cells in the center, and you can see that the PFAS does close these gap junction channels. And then on the right, we have cells pretreated with H89, hence closing the PKA pathway, followed by a treatment of the 6-carbon PFAS. Because these gap junction channels stayed open, it allows us to conclude that the only way the PFAS were closing the gap junction channels was through the PKA pathway. So that leads me to my second experimental question, and that would be, does the natural compound resveratrol found in red wine and peanuts prevent PFAS from closing gap junction channels? So we found that it actually did. You can see we have our PFOA and our PFOS controls, and then cells that were pretreated with resveratrol and then exposed to PFOA. PFOA was not nearly as effective in closing these gap junction channels. I will say resveratrol pretreatment was semi-successful in preventing PFOS from closing gap junction channels, but not nearly as much as the PFOA. My next steps would be to experiment with the effects of resveratrol pretreatment on the 6-carbon chain PFAS, as PFOA and PFOS are both 8-carbon chains, and then try to do some dose and time-dependent experiments to determine if we could get this prevention even greater, even higher. I'd love to raise the ability of resveratrol to prevent PFOS toxicity. So in summary, PFAS are forever chemicals, and the exposure is widespread and a current issue. PFAS pose a tumorigenic threat by dysregulating gap junction intercellular communication. Perfluorohexane sulfonic acid is likely closing gap junction channels via the protein kinase A pathway, and the natural compound resveratrol could potentially prevent PFAS tumorigenesis. And then finally, I just have some acknowledgments from my lab director, Brad L. Upham, and replicate experiments done by Jamie Leibold and Abdikenny Sheriff. Thank you.